All right, so here you can have one of the band. This is not your pen that I got from the lounge. East Everest High School band. This is not your pen. There are 3 of them in the box that said free, so I took them.
Oh, yeah.
But they said free, so I figured that really didn't mean that it wasn't my pen. It felt like it was my pen then. All right, so we don't want to spend all day talking about all of these problems. That was your homework. We do want to hit on a few of these things, though, because there's some important things that we really want to make sure you understand, as well as some kind of common questions. So this unit, we have 3.5 weeks before break, is 3 sections long. Okay, so this is section 1, talking about in general something called the cell cycle. There's 4 phases of the cell cycle: G1, S, G2, and M. Okay, on page 2 of this, it talks about in a 24-hour period, the M phase is about 1 hour. Okay, so section 1 is talking about the whole cell cycle as a whole. The next section we're going to talk about is about the M phase and the M phase only, which is called mitosis. Then we're also going to go through and we're going to spend some time on section 3 on an event called meiosis. Okay, so that's what this unit is really consisting of. We want you to know what the major events of this diagram are. We want you to know what happens in G1. We want you to know what happens in S. We want you to know what happens in G2 as well as in M phase. Okay? So, Mr. DeBoer has these sections kind of highlighted, and what he's going to do is he's going to write the main events. It's also summarized in Model 2. I know that you already wrote these things and some were already filled in for you. It's a good spot to draw it on this picture like he's about to do too. So, G1, S, G2, M. That's the sequence in which these orders, the phases go, right? It's a cycle. It repeats itself. It goes over and over again. It repeats. It's a cycle. So if we were to follow from that cell that has a star, and we follow through, that's the cycle. It goes G1, S, G2, M. Okay, and there's 4 phases here. And in G1, if we look at that picture, that cell is about this big, okay? And at the end of G1, it's about this big. Okay, now obviously I'm not exactly to scale. But what happened during G1 is the cell grows. That is the key event that happens during G1, is the cell will grow in size. The cell grows in size. Then what happens during S phase? S means synthesis, and we talk about photosynthesis, and photosynthesis is where we make. And what's happening is DNA is synthesized, or DNA is replicated, is what's going on in S phase. So what's going to happen? Cell cycle. We start with 1 cell, we end up splitting, and we have 2 cells. What those 2 cells need is they need everything that a cell needs in order to function. It needs organelles, it needs DNA. So the DNA gets copied during the S phase. Okay, the DNA gets copied during the S phase. Because during M phase, the cell's going to split. And half of the genetic information is going to go in one, I should reword that. 1 copy, that's better. 1 copy of the DNA is going to go into 1 cell, and the other copy is going to go into the other cell. So when we're done, each cell will have a complete set of DNA. And it's a genetic copy, identical copy genetically. Okay, so that replication occurs in S phase. During G2, we sometimes say that the cell prepares for division, or the organelles replicate. Okay, those are kind of the 2 things we often talk about during G2. Is the cell prepares for division, and the organelles replicate. Now, the Model 2 doesn't fit perfect with the organelles replicate, but whatever. And then during M phase, that's mitosis. The cell divides, and we end up with 2 genetically identical cells. They have one complete set of DNA, and they have a complete set of organelles that are gonna go through. If we start with one cell, we end up with two genetically identical cells. That's what happens during mitosis. Okay, that's what the cell cycle is. This is what's happening in your skin, this is what's happening in your bones, this is what's happening in your blood, this is what's happening in your heart, this is what's happening almost everywhere in your body.
What does the phase G2 say?
It says cell prepares for division and organelles replicate. Okay, so this is the cell cycle. This is occurring all the time in your body. Why does a cell divide? Yeah.
Where would we put G0?
Where will we put G0? G0 is something we ask you to label a little bit later, but it will go off of G1. And we can put it in right now, that's fine. So G0 is a resting phase. I don't remember what question they talk about it in, but they do ask you to put in G0. G0 is a resting phase in which some cells will go and they'll pause momentarily, sometimes or permanently, and they stop doing the cell cycle. It happens during G1. If it were to happen during S phase, there'd be an extra set of DNA and that'd be bad. If it did it during G2, it'd be too many organelles and that would be bad. If it did it during M phase, it'd be somewhere in between the division and that would be bad. So G0 is a resting phase and it goes on during, it happens, a cell will exit G1 to go to G0. Sometimes, depending on the cell, sometimes the cells will re-enter G1 and go through the cell cycle again. But sometimes a cell will go to G0 and will stay there forever. Kind of depends on the type of cell we're talking about. But G0 comes off of G1. Is a resting phase. Good question. Okay. Because there was a, somewhere we asked you, I don't remember where it is, to add that to your diagram. All right, so if we look at question number 3, during which phase, bless you, did the cell increase in size, said it G1?
21.
Oh, it's 21, okay, yeah. Question number 21 asked you to add G0, so there, that's where it would be. So question 3 says, in which phase does the cell increase in size? G1 is when it gets bigger. During number 4, during which phase does the number of cells increase? Well, we went from 1 cell at the beginning of M phase to 2 at the end of M phase, so during M phase is when the number of cells increase. So keeping that in mind, question 5 is asking, basically, why are you taller today than you were 3 years ago? Why does an organism grow in size? Well, there's 2 things that happen. One cell at the beginning of N phase is, we'll say, this big. Is this big. Okay? What happens to that cell during M phase? It divides in half. One cell was this big, it divides into 2 smaller cells. But there's twice as many cells now. So why are you taller today than you were 3 years ago? Because you physically have more cells in your body. Okay? However, these cells right at the end of M phase are small. One piece of paper got ripped in half, it's still the size of combined of one piece of paper. That in and of itself isn't why you are taller today than you were 3 years ago. But what happens to these 2 pieces of paper during G1 is those cells grow. And those cells grow, and they grow, and they grow, and now those 2 cells are fully grown, and they take up twice as much space. So the combination of during G1, the cells physically get bigger, and during M phase, we physically have more cells than we did 3 years ago. You do. That's why you're physically taller than you are today, or why you're physically taller today. Now, at some point, you stop growing tall. Okay, some of you may have hit that point, some of you still may be growing. Mr. DeBoer and I are getting, we're not quite there, to the point where we're gonna start growing shorter, okay? Because as you get older, that's hopefully quite some time from now.
I shrunk a little bit.
Did you really?
Yeah.
Huh. See?
Smidge.
What's that?
Smidge.
Since, like college kind of thing?
Yeah, late college, early pre-college,
Yeah.
but, or post-college, but yeah.
Huh. They talk about actually, if you, measure yourself in the morning, you're slightly taller than you are at the end of the day. You ever heard that?
Mm-hmm.
It's because your cells in your spinal cord with like just standing upright, they actually push down on you. They kind of compress a little bit during the day, and you get a little bit shorter. And then when at night,
Gravity.
right, they're able to kind of go back to their normal spot. There was a set of twin astronauts. Gosh, I don't remember their name. It wasn't too many years ago. One of them spent a chunk of time in the International Space Station, and when he came back to Earth, there's a picture of them standing side by side, and the one who was in space, I think he was in space for like 9 months or a year. He was out there for quite a while. He's physically taller than his twin because for like 9 months he didn't have gravity pushing on his bones and compressing those cells. They talk about in some video I saw that it won't take too long and he'll be the same size.
Yeah, I was gonna say, but
Mhm. But gravity's gonna compress those cells again, and they'll probably compress equally back to where they were before he went into space. But yeah, but you physically have more bone cells right now than you did 3 years ago. Mr. DeBoer and I are at the points in our life where we're maintaining bone cells. So for every 2 that are made, 2 are destroyed. Okay, so and I forgot to mention it, why does a cell go through cell division? There's really 4 reasons: to grow, to repair damaged cells, to replace like dying cells, repair because you got like a cut or damage. You break a bone, like you break your arm, it'll go through cell division very rapidly to heal that bone. So that's that repair piece. That's why you get that calcification, that bump on it, and reproduction. Okay, so some organisms will reproduce asexually, where one organism can divide and make 2 genetically identical. We'll get into that. Also, through the process of meiosis, which is still cell division, is the combination of egg and sperm cells, or production of egg and sperm cells. Okay, we'll get to, yeah, it's still coming. Number 6 and number 7, we're talking about cancer. So cancer in and of itself is something that we're all has impacted us in some shape and form in our life. Okay? The basic idea of cancer is this uncontrolled cell growth or uncontrolled cell division. Something's going wrong in this cell cycle, so your body starts producing more cells very rapidly and they're slightly misformed. Okay, something's not quite right about it. Okay, so when we talk about number 6, cancer is uncontrolled cell division, they talk about a mass of cells called a tumor. Now there's 2 different types of tumors. They often classify things as benign or malignant when they talk about tumors, and whether it's cancerous or not cancerous. But some cancerous tumors consist of cells that are much smaller. So they didn't grow to their full size. So if they didn't grow properly, what phase did they not grow in? Well, that would be G1, right? Because in G1, that's when cell growth occurred, was in G1. Okay. Now, number 7 talks about if the arrows represented in that model 1 is like the time it takes for a cell to divide, or if we peek down on model 2, they have some times listed for us. Okay, and in the model 2, they say, like, of all the hours, you add up the G1, S, G2, and M, it says it adds up to 24 hours. You have G1, which takes up 11 out of the 24 hours, about half, right? About half of the time a cell is alive or goes through its cycle, half of it is in G1. If G1 all of a sudden is shortened or removed, what that means is a cell cycle almost got cut in half. So instead of one cell taking 24 hours to make 2 cells, one cell is maybe taking like 12 hours or 13 hours. So they start producing cells much quicker. Now your body will destroy cells. They'll like, okay, we just made 2, here's some bad ones we're replacing, and we'll get rid of them. But if you start producing cells at a faster rate, then your body gets rid of them, and then you start to accumulate these, and that's kind of where that idea of tumors, these massive cells. Okay, so that's what kind of cancer in a nutshell is. We'll talk about it a little bit later. of these 4 phases, 3 out of these 4 are called interphase. So when we get to the next section, we're going to be talking about, hey, there's interphase, prophase, metaphase, anaphase, telophase, but interphase is G1 as in G2. Out of a cell cycle, 23 out of 24 hours is spent in interphase. Most of the cell's life is in interphase. And what is a cell doing? It's doing its normal stuff. If it's a heart cell, it's beating. If it's a muscle cell, it's contracting. If it's a bone cell, it's supporting you so you don't like fall on your face. Okay. So key processes in G1, cell grows. During S phase, DNA replicates. Those are the key things that happen during these events. And we can kind of see that, right? So in between G2 and mitosis, the number of organelles went from 600 to 300. That's cell divided. Half of the organelles are in each cell. During M phase, the DNA, during S phase, we started with 1, we ended with 2. During M phase, we started with 2, we ended with 1. The genetic information is split in half. There's 1 complete set in each cell. If we jump ahead to number 13, we will see number 13, we'll see that word throughout our upcoming section called cytokinesis. And when they break the word down nicely for us, the word cyto means cell, and kinesis means breaking or cutting. So when we talk about cytokinesis, cytokinesis is the physical division or the cutting of the cytoplasm. It's when the cells actually get chopped in half. That's cytokinesis. It's the division of the cytoplasm, or when the cell gets cut. 16 and 17. We want to talk about 16 and 17. So 16 and 17. 16 says that according to Model 2, it takes 24 hours for a cell to divide. Okay, it takes 24 hours for a cell to divide. So if we start with one cell, 24 hours later, one cell will have divided into 2 cells. Okay? Now, 17 says, what if if another 24 hours has gone by? So we start with one cell, it goes through cell division, we end up with 2. If another 24 hours goes, each cell will have gone through cell division, so that those 2 cells will each divide and become 4 cells. 1 becomes 2, 2 becomes 4. We call that exponential growth. Then 4 would become 8, and then 16, and then 32, and then 64, and 128, and 256, and then 512, and 1024, and so on. And if your body didn't destroy some of those cells, you would grow exponentially in height. You know, you get some big growth spurts, but you don't grow exponentially. And the reason for that is, as your cells are producing, they're always getting rid of stuff. Now, what happens if you cut your arm, and it's a pretty bad cut, your body says, fix that! And it'll kick it into the cell cycle very quickly. And it'll say, let's make as many cells as we possibly can as quickly as we can to try to fix that. And it doesn't get it perfect, and sometimes we have a scar because of that. The cell cycle didn't make perfect copies, so sometimes we get these we all have some scars from accidents in life, and it's because your body tried to heal it and heal it very quickly. 19. Starfish. If a starfish gets damages a limb, it'll often regrow a new one. If an adult sustains damage to his or her spinal cord, mobility is often impaired. They're paralyzed, right? That's what we're talking about. If a gecko loses its tail, it may grow a new one. Which type of cell is less likely to go through the cell cycle after being damaged? So that's the idea of that G0. So a cell will go through, once it's gone through its process, it may go to G0 and rest. It'll hang out there and do its stuff, its everyday functions. But sometimes some cells can come out of G0 and some can't. So which one is less likely to re-enter the cell cycle? Well, it's going to be our spinal cord, or our nerve tissue. If a starfish can regrow a limb, that means it will re-enter and start making new cells. If a gecko loses his tail and it regrows a new one, it will exit G0, go back through the cell cycle and grow a new tail. It'd be kind of cool if something happened and I lost an arm, that, hey, a couple years later, look, I got my arm back. It's not how it works for us. Same thing with the spinal cord. Like, we can cut our hand, we can cut our arm, we can have some pretty severe cuts, and our bodies will heal it. We can break a bone and our bodies will heal it. However, we can't heal like nervous tissue or spinal cords or brain cells. If those get damaged, they're damaged forever. Okay, so that's why they often talk about wearing helmets because head trauma and stuff like that. Okay, number 20. Occasionally, cells stop dividing and enter another phase called G0. If you damage your liver, new liver cells can be produced to replace up to 75% of the liver. However, if you sustain brain damage, your body does not produce new brain cells. Explain this observation using what you have learned about the cell cycle. So the idea is the brain cells go into that G0 phase, that resting phase, and they never come back out. However, nerve cells can go into G0. Liver cells can go into G0, and then what will happen is they will go through, and they can re-exit, and they can re-enter this, re-exit, that was the wrong word. They can exit G0 and go back into G1, and then they can replicate and regrow, which is kind of cool if you think about liver, like a liver transplant. You can go through and you can donate a portion of your liver to someone, and that portion of liver will regrow in someone else to be a full-size functioning liver, which is pretty cool. It also means that you as the donor can only lose a portion of your liver, and your body will also regenerate the rest of that liver. That's not true with like your kidney, not true with your heart. You can't cut out like half the heart and say, here, don't worry, I'll just grow a new one or a new part of it. But your liver can. I think we're getting pretty. Oh, and then 21. We already drew 21. We drew G0 back in Model 1. And I guess the very last question is 22A that I want to talk about. So if we consider a cell in G0, the resting phase, use information from Model 1 and Model 2 to follow. In order for a cell to divide normally, what would need to happen? So if a cell is in G0, they're in this resting phase, they exited G1, they're just kind of doing their everyday thing. If it were to divide, it would have to re-enter G1 to complete that cell cycle. Did you go through the division process? Okay? Right here on this stool, or on that lab table, there, right next to that wheel, there is a piece of paper. It's also linked, this is page 5 and 6 as the PDF that was linked on this. We'd like you to grab one of these. We want to just kind of touch on a few things dealing with cancer and radiation that kind of are an extension. So get up and either grab that from that stool or from that table, or on Canvas, it's page 5 and 6 of the PDF that we were working through too. All right, so this diagram on this new page looks very similar to what we see on the first page of the other thing we saw. The only difference is now we have ultraviolet light is shining on the cell. Where does ultraviolet light typically come from? Where do we normally get a dose of UV light from? The sun, yeah, the sun. Okay, so if we're out in the sun, we're getting some ultraviolet light or some ultraviolet radiation. And there's a lot of different types of radiation. Is kind of how we generally classify. Technically, they're electromagnetic spectrum, anywhere from radio waves, like you listen to the radio on the way here, to microwaves, to X-rays, to gamma rays and stuff. And different ones have different amount of energy levels. And Roy G. Ebert, red, orange, yellow, green, blue, indigo, violet, okay, in the color spectrum. The very next wavelength is ultraviolet. And ultraviolet is the threshold. It's the like kind of the first type of wave that has enough energy that can cause cell damage or DNA mutations. Okay, it possesses enough energy that it can zap your DNA and make a mutation or just screw up your DNA. We call them mutations. So if your DNA gets mutated, something happened. Now, sometimes those mutations make absolutely no difference to the organism. You wouldn't even know they got mutated. Sometimes it's maybe a beneficial mutation. Maybe now you can see in the dark. That'd be a really kind of weird mutation, but maybe you get a mutation that allows you to have night vision. That could be kind of a cool mutation. Don't count on laying in the sun for too long, getting a sunburn, and all of a sudden you start seeing in the dark. That's not how that works. But some mutations are good mutations. Some mutations make no difference, and some mutations are bad. Not all mutations are bad. They aren't all sci-fi. You got that third eyeball on your forehead kind of mutation. So if a mutation were to occur, what could happen? Well, this DNA could, the UV light is getting blasted on the cell in G1. Now what happens during S phase? The DNA is replicated, it's copied. So if the DNA gets mutated or screwed up in G1, and it goes through S phase, what that means is that mutated DNA just got replicated and gets copied. And if that cell were to continue through the cell cycle, what happened is that one cell with that mutated DNA just divided, and now you have two cells with that mutated DNA. Okay? So if that DNA is mutated, in S phase, the mutated DNA will be replicated or copied. And then if it goes through and divides, again, we end up with two cells that have mutated DNA. Now, sometimes it talks about this in this, read this, the cell cycle has a regular system of checks and balances that prevents damaged or mutated cells from proceeding to the next phase. So most of the time, if something is mutated, our body catches it and says, hey, that's messed up, stop, don't make that new cell. But it doesn't always. One way an organism deals with this problem is to kill the damaged cell before it passes this problem on to its daughter cells. This normal process is called apoptosis. It's basically where some normal cells are, they're destroyed. Okay, we destroy some of the cells. That's pretty typical. Apoptosis is a thing.
Apoptosis.
Apoptosis, thank you, apoptosis. So we'll go through and say, hey, something's not right, destroy that cell, therefore we don't make that mutated copy anymore. However, that's not always the case. So for 26, why might it be beneficial for an organism for damaged cells to enter G0 instead of dividing? Because we aren't going to replicate that mutated cell. Okay, that's the idea. But what sometimes happens is number 27. What could happen after several cell cycles to organisms whose damaged cells did not go through apoptosis? In other words, what if a damaged cell that is supposed to die does not? And the idea is your body can start producing something like a tumor, which is referenced before, or cancer. Because what cancer is uncontrolled cell division. Okay, it's uncontrolled cell division. So if your cell doesn't go through apoptosis, then we can start forming cancer cells. So how do we treat cancer? Well, there's three main ways doctors will treat cancer. One way they can treat cancer is they physically try to remove those damaged cells. If we physically cut out the cells that have that mutation in it, they are no longer in your body to replicate and to make those new mutated cells. So if they physically cut out and remove that section, in theory, your cancer or that tumor is gone. A lot of times though, with cancer treatment, they'll do that followed up by either radiation or chemotherapy. So they'll try to get all the cells out first, they'll try to physically remove them. Then what they'll do is they'll try to treat any other ones that are left. So when we turn this over, we're going to skip to question number 29, and we're not going to worry about the other ones. But 29 is dealing with radiation. So some types of cancer are treated with radiation, similar to ultraviolet light. kind of like ultraviolet light, but it's got a lot of energy. So why might it be beneficial to irradiate cancer cells? Well, the idea is we can go through, and just like UV light can cause your DNA to be mutated, they can take radiation and they can focus it in a specific spot, and they can either force the cell to go into G0, which then hopefully once it goes to G0, apoptosis will occur, and then any cells that are left over will die, they won't replicate. Okay, that's ideal. Okay, is any of the cells that are still around will then basically just get shut down and they won't replicate, or they'll physically kill those cells. The radiation can physically like break down the cells so they don't function, it'll kill them. And if that cell's not alive anymore, they aren't going to replicate. Okay, so radiation can be used to try to either force a cell into G0 or to physically kill those cells so they don't replicate. 31 is referencing something called chemotherapy, and we all have heard of chemotherapy, and a lot of times we often hear about the side effects of chemotherapy, and the side effects typical with chemotherapy are hair loss and like nausea. People are getting really sick from chemotherapy. So what chemotherapy is basically a drug that goes through the body and it looks for rapidly dividing cells. Because what is a rapidly dividing cell? That's cancer, right? So this chemical goes through the body, and I don't know the mechanism and how this works. I'm not gonna lie and pretend like I understand how chemotherapy works. But I know that it looks for rapidly dividing cells, which is cancer. So it goes through, finds those rapidly dividing cells, and it shuts them down. It kills them, it destroys them. That's good. It's killing the cancer cells. However, by default, it also kills some other types of cells. Because in your body, we have naturally occurring cells that are rapidly dividing. Your hair follicles, why does your hair grow? Because right at the base, there's cells that are rapidly dividing to push out new hair. So what happens is chemotherapy sees rapidly dividing cells at the base of your hair, and it kills those too. That's why hair loss is pretty common with chemotherapy. Because those are rapidly dividing cells. And chemotherapy doesn't differentiate between cancer and hair cells. Same idea with your stomach lining. You have this mucus lining in your stomach that is rapidly dividing. It's this very harsh environment, so they're constantly replacing cells in your stomach. Same idea is that mucus lining gets kind of worn thin, which then leads to upset stomach and nausea that is associated with chemotherapy as well. Chemotherapy and radiation, like I said, are kind of follow-ups, or sometimes are follow-ups to removal of surgery. Depending on the type of cancer, depends upon how fast it spreads, it affects the treatment options, it affects survival rates. So oftentimes they'll try to treat it as quickly as possible to try to prevent it from spreading to other parts of the body. That's chemotherapy, radiation in a very overview. Simplified, thank you, in a very simplified fashion.
Anything you need back up? I tried to not go too fast as far as my notes. Everybody get what you need?
All right, we are going to wrap today up with a 9-minute video on the cell cycle. Only took 4 times. What? Today's 4 for me.
been sitting in class and thought to yourself, I wonder what my skin cells are doing right now at this very moment? This kind of pondering may be unique to me, maybe, but wouldn't we at some point wonder what our cells are doing right now? Because if you remember, as part of the cell theory, you are made of cells. All living things are made of one or more cells. Many multicellular organisms like you have cells that work together, working together as part of a body tissue. Body tissues working together as part of an organ. Organs working together as part of an organ system. Your cells are specialized to work in these different levels of organization. You have skin cells, stomach cells, muscle cells, just to name a few, and their functions need to be regulated. These cells actually are regulated as part of something called the cell cycle, and that is going to relate to my question of, I wonder what my cells are doing right now. Cells themselves can grow in size, but let's put it in perspective now. A multicellular organism isn't growing because each individual cell is getting bigger. A multicellular organism itself grows by making more cells, by the cells making more cells by dividing. That's cell reproduction. One reason that you're bigger than you were when you were 5, unless you are 5, is because your cells have divided to make more cells. Mitosis and the cytokinesis that follows, that splits the cytoplasm, allows you to make new body cells. But you don't want that cell division happening all the time. Why? It is likely that you've heard the term cancer before. We have had family members that have battled cancer before. It is definitely a relevant topic for all of us. Cancer is in part due to cells that divide too frequently. The cells are not regulated. They are uncontrolled. Cancer cells can have other problems too. They might not be able to communicate with other healthy cells. They may not be able to carry out normal cell functions. They may not securely anchor themselves like other cells do, which can make them more likely to travel somewhere else. Some cancer cells have the ability to secrete their own growth hormone that makes blood vessels divert over to those cancer cells and supply the cancer cell with nutrients, which can take nutrients away from healthy cells. Why do cancer cells become this way? Well, there's a lot of research in this area. With some cancers, there may be genetic links, making some cells more susceptible to having problems. These genetic factors might run in families. Exposure to toxins, radiation, excessive exposure to UV light, all of these can be risk factors for some cells to become cancerous. The uncontrolled growth that cancer cells have gives rise to more cells like them, which can develop into a tumor. Some tumors stay put, but some do not. Now, the good news is that scientists continue to develop better treatments, which include destroying the cancer cells with radiation or medication, such as chemotherapy, which will target cells that divide frequently. Maybe someday you will be part of helping to meet the challenge of trying to eliminate cancer, because the fact remains that these cells are not participating in the cell cycle like they should. So what is the cell cycle? The cell cycle is often represented as a pie chart like this. Cells are either in one of two different phases. A phase called interphase, where the cells themselves are growing, replicating their DNA, doing their cell functions, or they are in M phase, which includes mitosis and the actual splitting of the cytoplasm, cytokinesis. So this M phase is where cells actually divide to make more cells, but cells spend most of their time in interphase. So most of the time, they're not dividing. Now, depending on what kind of cell, it might do mitosis more or less often. For example, your hair follicle cells do mitosis frequently, which is why your hair can grow at the rate that it does. It's also why many cancer drugs may also target hair follicle cells, because many cancer drugs go after cells that do cell division frequently. It's a big deal for cells to hit this M phase. If a cell has an error, a harmful mutation, for example, you do not want it to divide, because then it will create another cell that has this same issue. That's where checkpoints come in handy. Along the cell cycle, there are checkpoints to check that the cell is growing well, and replicating its DNA correctly, and doing everything it's supposed to do correctly before it divides. To better understand those checkpoints, let's further divide the cell cycle pie chart. We have G1, S, G2. All three of those are part of interphase. Then we have M phase, where mitosis will happen. During G1, the cell individually itself grows. Then it replicates its DNA in S phase. You can remember that because the S is for synthesis, which means to make something, and it's making DNA. Then G2, the cell grows some more in preparation for mitosis. So let's take a look at checkpoints. We've got one here in G1. This checkpoint checks, is the cell growing well enough? Is its DNA damaged? Because if it is, you definitely don't want it to move on to S phase where it would replicate DNA. Does the cell have the resources it needs if it were to keep moving on? This checkpoint in G2 checks if the DNA was replicated correctly back in S phase. Is it growing well enough? Does it have the resources it needs to continue? Okay then, moving on. This next checkpoint in M phase is my favorite checkpoint. It checks in the stage metaphase to make sure the chromosomes, which are made of DNA, are lined up in the middle correctly, that they're all attached to the spindle correctly, because if they're not, the chromosomes will not be separated correctly. So now you may have two big questions. First, what happens if the cell doesn't meet the requirements of the checkpoint? And second, what is doing the regulating of this cycle anyway? To address the first question, if the reason the cell can't go past the checkpoint is a reason that can be fixed, the cell may kind of pause here until it can fix that issue. But if it can't be fixed, then the cell does something called apoptosis, which basically means the cell self-destructs. This ensures that a cell that is damaged beyond repair will not go on to divide. So what is doing the regulating anyway? We've mentioned before that proteins are a big deal. Genes in your body can code for proteins that do an assortment of functions, and there are many proteins involved with regulating the cell cycle. Some of them are positive regulators because they allow moving forward in the cycle, and some are negative regulators that might make things stop. The proteins themselves can be sensitive to cues inside and outside of the cell. So, for example, two proteins that are involved in positive regulation are cyclin and CDK. CDK is specifically an enzyme protein, a fancy kind called a kinase, which is worth a Google. CDK can have different forms of cyclin protein bound to it. Different types of cyclin rise and fall throughout the cell cycle. And the rising and falling is based on a variety of signals to determine when the cell should move on to the next cell cycle phase. Typically, each cell cycle phase, G1, S, G2, N, will tend to have a different cyclin binding with the CDK. The rise and fall of cyclin types and the role CDK has when it's active is a fascinating subject to explore. Remember that vocabulary word we said, apoptosis? Proteins that are negative regulators, for example, a protein called P53, can be involved in initiating apoptosis. Again, we encourage you to explore beyond the video. One last thing to mention. There are some cells that don't go through the phases we mentioned because they're actually in G0. That's a 0, by the way, and not a no, because if it was a no, then it's a go, and G0 is kind of the opposite of that. G0 is a resting phase. Now, cells here are still performing cell functions, but they're not preparing to divide. Some cells go here temporarily, maybe if there's not enough resources around, for example. But some, like many types of neurons in your brain and spinal cord, may stay here permanently. If they stay here permanently, they'll never get to M phase, so they will not divide. This can be one reason why a major injury to the brain or spinal cord can have challenges with healing, as many of those cells may not be able to replicate, a topic that definitely continues to be researched. Well, that's it for the Amoeba Sisters, and remind you to stay curious.
All right. So, tomorrow, we gotta get that out. Tomorrow, virtual day, you have guided notes to work through on cell cycle, interphase, prophase, no, not yet, interphase. And they are right there, paper copies are right there. If you so choose to do a paper copy to submit via Canvas, there it is. If you would like to mark up a PDF, it is linked as a PDF in Canvas. If you want to write on a Google Doc, that's also linked in Canvas. But your homework for tomorrow on your virtual day is guided notes. So there's a stack right there if you want a paper copy, or there is a stack right there if you want a paper copy. Once you grab those, if you want a paper copy, wipe off your desk, and we'll see you guys the next time.
Have a marvelous